This is a video abstract for the PhD proposal titled Nanomaterial-Enabled Disposable Diagnostics for Rapid Point-of-Care Detection of M. tuberculosis by Kristin Byers. Tuberculosis, or TB, is a widespread infectious disease caused by the bacterial strain known as Mycobacterium tuberculosis. The World Health Organization, or WHO, reported that 1.5 million people died from TB during 2013, and 360,000 of those individuals were HIV positive. Worldwide, 9.6 million people are estimated to have fallen ill with TB in 2014, and 5.4 million men, 3.2 million women, and 1 million children globally have fallen ill with this disease. To reduce this burden, detection and treatment gaps must be addressed, funding gaps must be closed, and new tools must be developed. In 2014, 6 million new cases of TB were reported, and fewer than 2/3 of the 9.6 million people estimated to have fallen sick with the disease felt treatment. Worldwide, 37% of new cases went undiagnosed or were not reported. The quality of care for these people in the latter category is not known. Mycobacterium tuberculosis is difficult to identify due to its persistent nature and slow development, and because of its ease of transference, rapid and sensitive detection is critical for the control of tuberculosis. Worldwide, 9.6 million people are estimated to have fallen ill with TB in 2014. To reduce this burden, detection and treatment gaps must be addressed, funding gaps must be closed, and new tools must be developed. TB is difficult to identify due to its persistent nature and slow development. Because of its ease of transference, rapid and sensitive detection is critical for the control of tuberculosis. In regions where medical care and transportation are scarce or unreliable, the need for one-stop point-of-care diagnostics is imperative. These diagnostic needs can most effectively be met by the use of a biosensor. Although products do exist and laboratory demonstrations prove that tuberculosis can be detected with a biosensor and with other products, the issues with time and sensitivity are a major problem for this blah blah blah. The goal of this project is to create a highly selective nanomaterial-enabled biosensor for the detection of M. tuberculosis that is feasible for use as a disposable diagnostic tool in the field. Objectives 1 through 3 specify how this will be done and how it will be tested. More specifically, a paper-based disposable diagnostic platform will be created using graphene nanocellulose. At least 2 aptamers will be sequenced and thiol tagged, and the aptamers will be immobilized onto the metal decorated graphene paper. All sensors will be tested in the laboratory for baseline selectivity, specificity, limit of detection, response time, and hysteresis using clinical pathogenic M. tuberculosis and buffer. For control purposes, another electrode without an aptamer will be used as a reference in all experiments. All measurements will be verified with standard analytical instrumentation used to detect TB in hospitals. This project will improve upon existing point-of-care technologies for the sensing of TB, and the intellectual merit of this project is to develop a new approach for fabricating rapid, low-cost disposable biosensors for TB that can be integrated into point-of-care field devices for the detection of AIDS and other issues.